You get treated. So,
That's a good idea. I like that answer.
There's nothing better than like when you have to go to the crunch.
Because it was water. I had no idea to see like if I don't or they'll find me. Maybe it's not Gary said he did last week.
probably could have never had that for that
That would be worse.
Hallelujah.
The only thing that was open was men.
So what it is someone's coming in the day before. Yeah. So I'll just Microsoft really had one of those connector buttons.
Right.
They don't need
Good.
That's true.
We're picking the first day.
Were you able to do your charts yesterday?
You see me here.
I couldn't have it down.
Well, there's some, it still doesn't fill in on the site. It's all, they'll give it, they won't charge next week.
No, they're not. No. You know what? No, they can't.
Just waiting to make sure we have.
So you know what? That's right.
You know we do, Drew?
Trixie, yes. That's thinking we do.
Oh, okay.
No, I know.
Okay, let's go ahead and get started. Drew tells me the sound is operating correctly. Mike Weiss, if you're listening, if you want to text me to confirm that you have good audio, that would be another confirmation. All right, the word of the day is Washington, and the speaker of the day is David Jeong. He's going to talk about the status of food allergy desensitization. I don't know if he's going to mention the clinic that is starting up at Children's, but for anybody in the community or anybody listening wherever, the clinic for doing food challenges has started, I think, on a Q2 week basis. Frank, is that right? At Children's. And a number of our, Mike's wife says the sound is good. Thanks, Mike. So, David, why don't you go ahead with your presentation?
Okay.
Thank you. So I'm going to talk about the current status of food allergy desensitization. Hot topic these days. Those are my disclosures. The outline that I'm going to follow today, I'm going to talk a little bit about the impact of food allergy on patients and also sort of on a broader scale, to kind of provide the backdrop for why we're doing all this. I'll talk specifically about OIT, Slit, and patch therapy, and then we'll come together and just kind of tie it all together and kind of figure out exactly where we are with all this stuff. So food allergies, not a hard sell to this crowd. It's an increasingly prevalent public health problem, significant adverse medical, psychosocial, and economic impact. We know the prevalence overall across all ages is 3 to 4% in the United States, which equates to about 15 million Americans. Worldwide, up to 250 million individuals, so a very significant problem. Dollars, in the United States, we're seeing recent numbers up to $25 billion in terms of overall financial impact from food allergies. And what we're telling people to do is, avoid, be ready for expectant management with epinephrine, antihistamines, right? So how is that approach working out? Well, these are some numbers that they're not necessarily brand new numbers. They've been around for a little while, but they're still pretty impressive. So we're seeing that 50% of kids who are diagnosed with food allergies within 5 years of diagnosis experience at least one accidental ingestion. within 10 years, 75% of children experience an accidental ingestion. This is despite diligent avoidance. Almost 54,000 episodes of food-induced anaphylaxis annually, and so the food allergies have become the leading cause over the last 2 decades. 125,000 ER visits annually, that's in the United States, and potentially up to 200 fatalities from anaphylaxis per year. And most of those, more than 90% of those, are due to nuts. So just from that alone, we can understand why there's sort of a motivation to figure something else out. So in the last 10 years or so, there's been a high priority on proactive food allergy treatment, and specifically on immunomodulating therapies. A lot of different groups interested in figuring this out, from patients to families, researchers, advocacy groups, and people who have money. And in the last 5 years, there has been fairly significant progress. So that's really what I'm going to focus on today with all of the different modalities. So this is a general schematic that was published, I believe, a couple of years ago. It's kind of it's breaking down the sort of different food allergy therapies that are out there. Allergen-specific versus allergen non-specific. Today, I'm going to, as I mentioned, I'm going to focus more on this side of things. But important to note, particularly on the allergen non-specific side, Chinese herbs, that's something that's been studied a fair amount and seems to show some promise. Anti-IgE, Omalizumab, I'm going to talk a little bit about that today. And down on the sort of the preclinical side, particularly these modified products, the heat-killed bacteria through rectal administration, that's actually been studied very preliminarily and was actually shown to have fairly significant systemic reactions. So that's kind of not gaining the momentum that it once had or was once expected. And immunotherapy based on recombinant peptides and DNA sequences. Not a significant amount of momentum with these yet, but the ideas are interesting nonetheless. So focusing on allergen-specific immunotherapy, as I mentioned, a lot of focus on this in the last 5 years, particularly the OIT, Slit, and patch. This is a snapshot of what's ongoing or actively recruiting at this very moment. This is just information I got from clinicaltrials.gov. So right now there are 25 OIT trials, 4 OIT plus Omalizumab trials, 5 Slit trials, 4 patch trials, and they all have to do, they're all sort of using the DBV products. And then there is one SCIT trial with modified codfish protein that's been going on for a couple of years. That's despite the data that came out in the 90s with peanut SCIT. The idea there is using the modified recombinant protein to alter the risk. So you can see that the predominant focus here is on OIT. That's where we have most of the data at this moment. In terms of where these studies are going on, most of them are happening in the United States. That might be intuitive. There are a couple of other places around the world, France, Denmark, Israel, and Finland, where there are some smaller studies going on. And the foods that are being looked at with these trials, nuts, seeds, milk, wheat, and fish. Of course, there have been other foods that have been looked at in the past as well. All of these trials, whatever modality we're talking about, have a very similar design. There's an initial dose escalation day, and after that there's a build-up phase in which these patients are taking daily allergen, but their doses are being increased once every week or once every 2 weeks with the more recent trials, it seems like most of them are using a biweekly dose increase regimen. And then there's a maintenance phase where they're continuing a set amount for the rest of the protocol. So looking at all the papers, and there are a lot of papers now, there's quite a bit of heterogeneity in terms of the overall design, the dosing, the duration of each of these phases in the studies. So that lends itself to some difficulty in making solid conclusions. I'll get to that a little bit later. I'm going to sort of dive into OIT. OIT, as you know, is oral food administration, typically in the form of food flours. Dose ranges are usually 300 milligrams to 4,000 milligrams. Probably all know this. The very first published success with OIT over 100 years ago with egg anaphylaxis in this young boy, and it was actually successful, the protocol they used. And I've already mentioned that, this really has the biggest body of evidence in terms of the various immunotherapy modalities. So this is a schematic of the sort of mechanism of OIT and all these modalities. So with OIT, you're exposing allergen to gut dendritic cells, and that eventually leads to downstream signaling that upregulates Tregs, production of IL-10 and TGF-beta. And the thought there is that you have a downregulation of T effector cells, Th2 cells, and all of the cytokines that are produced. You have alteration of B cell activity and antibody production, so you have typically a reduction in IgE. Most of these trials will actually see an initial rise in the IgE and then eventually a decrease, and that can be over months or up to a year before you see the actual fall. And then also an increase in the allergen-specific IgG4. And then also you'll have a downregulation of these effector cells, mast cells, basophils, and eosinophils. So I'm going to highlight a couple of of studies that have been out over the last 5 years or so. The concept of baked milk and egg, that's not terribly new to us. I think we all are pretty much on board with this by now. These are the study on baked milk and baked egg, two of the earliest studies on this topic, and the basic conclusion from these studies is that approximately 70 to 80% of kids with egg and milk allergy can safely tolerate baked or heated forms of those foods, respectively. And then this study from Kim looked at, it kind of took it a step further in terms of actually incorporating baked milk into these children's diets, and found that those who were tolerant to baked milk were 28 times more likely to become tolerant to unheated milk compared to those who were actually reactive to baked milk. And those who actually incorporated it into their diet were 16 times more likely to outgrow their allergy relative to the comparison group. So there is an advantage of figuring this out, incorporating it into their diets if you can. Wanted to point out that there is an ongoing study through COFAR. They're actually trying to compare egg OIT versus baked egg treatment to see which one can be more effective in terms of resolving an egg allergy. And there's also a study in Israel looking at whether baked milk OIT can actually have any impact on unheated milk allergy. So those are both ongoing. Switching to peanut, so this is a study from 2011 from the Duke group. This trial had 28 kids. They looked at they split their groups between peanut OIT and placebo, and it was a year-long treatment. They were trying to reach 4,000 milligrams maintenance dose. They found that the active treatment group was able to significantly increase their peanut consumption. They tolerated a 5-gram challenge at the end of treatment as opposed to just 280 milligrams in the placebo. So there is certainly a strong desensitization effect. As far as safety, half of the subjects experienced reactions, just a little over 1% in terms of all of the doses, led to some symptoms. Only 2 of them required epi. And then for the rest, with the buildup and with the maintenance, there were far fewer issues with reactions. They saw in their patients in the treatment group smaller skin tests, reduced THT profile, increased IgG4, and increased Treg. So basically exactly like the schematic we looked at.
They exclude the most severe patients on that study.
Yeah, so that's an inherent issue with almost all of these studies that I'm going to talk about, is that's always the number one exclusion criteria, and that obviously leads to a lot of questions about what this actually, how applicable this really is. But yes, this group, they did exclude the severe anaphylaxis.
How, when you say the dose escalation day, did they go all the way to full dose in one day, or did they just do a half of it? How far?
No, they with this, so the actual, what they got to on the first day, I apologize, escapes me, but it's a very small fraction of the maintenance dose. Yeah, but I'll talk about some studies that where rush desensitization is being implemented. That's typically with Omalizumab. Nobody's really tried a rush desensitization without Omalizumab, and on those studies, you can get up to 25% of the maintenance dose on day 1. So this is the egg immunotherapy study from the COFAR group that came out in the New England Journal 2 years ago. A couple of highlights from this study. This was it was a double-blind, placebo-controlled study, 55 kids, and they intentionally chose the older kids, theoretically, to find the kids that have a lesser chance of outgrowing the egg allergy as opposed to the younger ones. And it was all based on clinical history and IgE levels. They did not perform baseline challenges to confirm the allergies. They randomized 40 to the treatment group and 15 to placebo, and the maintenance dose they were reaching for is 2 grams. So what these guys did is they challenged their treatment group at 10 months, after 10 months of therapy, and they found that just a little over a half of them passed a 5-gram challenge. They challenged the placebo group at this point too, and nobody passed this challenge. And then they did it again at 22 months, and they found that a few more, 75% of the treatment group passed actually an even higher challenge, a 10-gram challenge. So they are seeing a desensitization effect with this protocol. Lots of active doses, and approximately a quarter of them were associated with reactions, but most of them were oral itching. There were a few systemic reactions, and a couple of dropouts from the therapy as well. But most of them were primarily mild and oropharyngeal. And in the active group, they saw lower IgE, higher IgG4 at 10 months. Those were early effects, and then the later effect, at 22 months, they found reduced basophil stimulation, and skin prick actually started to get smaller. So this study brings, I think, is a good segue to this topic, desensitization versus tolerance. To remind you all, desensitization is a non-reactive state with a regular allergen administration, but the reactivity returns once you stop administering the allergen, as opposed to tolerance, where you can ingest allergen without reactions, even with periods of withdrawal. So there's a new sort of term that has been thrown around this year in the literature called sustained unresponsiveness. So this is, it's a similar sort of concept to tolerance, but applying it to these immunotherapy trials, it's described as the ability to pass a challenge after stopping OIT and then successfully introducing into the diet ad lib after you stop therapy. So this term came from, back to this egg study, it came from the egg study, and so it kind of caught on and everybody's been using it since then. So a couple of more highlights from this study. These guys claim to be, and I believe they are, the only double-blind placebo-controlled trial that is reporting sustained unresponsiveness in a subset of their patients. So just as a reminder, the 75% that passed the challenge at 22 months, they were told to stop, but they stopped therapy at 22 months, and they were told not to eat any egg products for 4 to 6 weeks. They admit in their study that they were not sure whether everybody in this group absolutely avoided eggs, but they tried. And then they were brought back 2 months later, and then they were challenged again to the same amount. And they found that 11 out of the 30 passed their challenge at that point after no egg ingestion. So hence a sustained unresponsive state. It's not clear based on the study what was specifically different about this group in terms of how they were able to become unresponsive, but they were. And they followed up at 30 and 36 months, and they were still eating egg ad lib without any problems. So mention here that so the placebo group was challenged at 10 months, but only one out of the remaining, or one out of the people in the placebo group was challenged at 22 months. And that was because that person's egg IgE fell to below 2. And it turns out after the patient was challenged that the person failed anyways. But that some may say is an inherent weakness or limitation of the study, because you still have, despite what their arguments are about their treatment group, you really can't comment about, is this truly a treatment success, or is this spontaneous resolution of egg allergy? It's hard to know, and that's going to be always an issue. Even if you do challenge your placebo groups, there's always that question.
Did those 11 have an elevated IgE still?
I believe they did, but I didn't. The IgE did fall after 10 months, but I don't remember seeing. I don't recall. I apologize about how much it fell, but it was still certainly within the positive range.
So the difference between tolerance and sustained unresponsiveness is just they're not sure this is going to last, so they make up an intermediary term.
Nice.
Yeah. So getting to peanut, this is another study that looked into the concept of sustained unresponsiveness. So this study is utilizing the same cohort as the kids in the Jones paper that was published in 2009. This was looking at 39 kids who completed 36 months of peanut OIT. So they only were able to get 24 out of the 39 who had actually passed the challenge at the end of therapy. So these 24 were advised to discontinue OIT. And they actually told the people who failed the challenge to continue on for a total of 5, so 2 more years. And so that, I don't believe that there's any published data on that group yet, but it'll be interesting to see what happens with them. But this group, the 24 who did pass, after stopping OIT and no peanut ingestion, they came back 4 weeks later and were challenged again. And they found that half of this group tolerated a 5-gram challenge and half did not. So that group that passed, they were told to continue eating ad lib, and there was follow-up out to 40 months, and they were still eating without any problem. So hence this group, sustained unresponsive. I don't think that there's any more data beyond 40 months, so I don't know what's happened to this cohort since, but a positive effect. And the group that, the other 12 that failed, their reaction threshold was significantly higher. In terms of median reaction threshold, all the way up to 3750 milligrams, as opposed to less than 50 milligrams prior to enrollment. So even the ones who are failing are showing still some positive effect. And this group also showing smaller skin tests. The IgG4 and Tregs did improve, but there was no significant difference between the sustained unresponsive and the reactive. And these authors stated that at the end of their study, the degree of elevation of the RH2 first and then the P9 IgE were the best discriminators for those who would fail and those who pass. So this is an ongoing trial. It's called the IMPACT trial. It's an ITN-sponsored trial. It involves 3 years of peanut OIT, and they're looking to shed more light on the concept of tolerance. These are all young kids. They're going to finish 134 weeks on OIT, so pretty long period of time, and then they're going to just stop, and they're going to be off for 26 weeks, which to my knowledge is the longest that any of these trials have or will implement in terms of testing tolerance. So they'll be challenged at the end of 26 weeks off therapy. So it'll be interesting to see what they find. So I'm gonna talk about Omalizumab. It's to me an interesting topic and could potentially add some benefit. So the idea there is that using it with OIT can help you desensitize patients more safely, more rapidly, and more effectively. And there is literature dating back to over 10 years ago, looking at Omalizumab with peanut protein and actually decreasing the dose threshold for reaction. And there's positive data there. And this last point here, this author showed that it could increase the threshold by up to 80-fold, so significant positive impact. But there wasn't, until just a few years ago, there really wasn't much data on Omalizumab and in the use of, or used with desensitization. So that obviously would be an interesting question to answer. So this Stanford group published this, I believe it was a letter to the editor, looking at Omalizumab pretreatment followed by rush desensitization in kids with milk allergy. And to my knowledge, this is the first one that actually was ever recorded. So this one was a small trial, 11 kids. The median milk IgE for this cohort was 50, a fairly broad range, 42 to 342. They were pre-treated with Omalizumab for 8 weeks. And then after 8 weeks, the rush desensitization protocol began. And these kids stayed on Omalizumab for 16 weeks total. So they found that 9 out of the 11 kids tolerated a gram of milk powder on the first day. So that is, it's a lot more than a typical protocol without Omalizumab. And I think with this one, the maintenance dose was 2,000. So they basically got to 50% of their maintenance dose on day 1. And then they built up, and they were able to get up to the maintenance dose of 2 grams in a median of 8 weeks, which is significantly faster than you would see without. And then at 24 weeks into treatment, they underwent a challenge, more than 8 ounces of milk, and all 9 who had initially reached that higher dose on the first day, they all passed their challenge at 24 weeks. Most of only about 2% of all the doses led to reactions, and the overwhelming majority of them were mild. Nothing. There were a couple of severe reactions, but they were definitely outliers. So overall, that is an improvement compared to trials without Omalizumab.
How many, David? What was the number of doses of Omalizumab that they got?
So it depended. The dosing was based on weight and IgE level. So there were some who. I'm sorry.
Total IgE.
Total IgE. Yeah. So they, so there were some who were getting it every 2 weeks, some who were getting it every 4 weeks, and the actual dosages varied with the subjects as well.
So did their IgE have to be under, like, the...
I don't, yes.
Okay.
Sorry.
You know how some kids with food allergies, their IgEs are 2,000 or 1,000.
Yeah.
They would be excluded from that study?
Right. So some of the later studies, they are using that cutoff. I don't believe that, to my knowledge, I don't think they use that cutoff for this milk study. But you're right. They're the later studies, so I'm going to actually talk about some of them right now, but some of the later studies, they are using a IgE of 2,000 as a cutoff. Yeah. So this is a study from 2013 from Schneider looking at Omalizumab in peanut allergies. Again, this was just a small trial, 13 kids, and they were actually looking at severe allergic reactions, but not too severe. A median peanut IgE of 229, a pretty broad range of IgE, and these one of the strengths of this trial is that these patients were all confirmed by baseline challenges. So slightly different protocol. They got Omalizumab for 12 weeks, and then they started their rush desensitization. And all of the 13 kids got to a goal dose of 500 milligrams, or cumulative 1,000 milligrams on the first day, with very minimal or no symptoms at all. And then out of the 13, 12 were able to reach the 4,000 in 8 weeks, just like the previous study. And so in these subjects, the Omalizumab, they didn't discontinue it until they reached maintenance dose. They were challenged 12 weeks off of Omalizumab again, and all 12 of these subjects tolerated 8 grams of peanut flour at that point. Safety-wise, very similar. 2% of all doses led to reactions. Almost 90% of them were mild and either not treated or treated with antihistamines. And I wanted to point out that of all these reactions, unfortunately, this poor one subject experienced 35% of them. And in this cohort, the IgE levels decreased a fair amount, but they were still pretty high, at a median of 70. So,
They were continuing daily peanut ingestion. There was no tolerance test here.
This one, there was no tolerance test. I think that the protocol was a year, but I don't think they did any tolerance studies after they finished. So it'd be interesting, with this particular cohort, if they had kept going, what would happen to the IgE. So that's most of the day. I'm going to talk about one or two more trials in a few minutes, but that's the extent of what we have with Omalizumab. So not very much. The pros, it does with the preliminary data we have, it seems to lower the incidence of moderate to severe reactions actually quite a bit. I didn't stress this too much before, but with some of the OIT trials, up to 30% of individuals experience potentially systemic or severe reactions. The shorter time to maintenance, most of the trials are seeing 8 weeks as opposed to months, and in some even longer than that. So the question is that shorter time, fewer doctor's visits, fewer dose escalations, is that going to justify the cost? I think that can be debated either way. The cons of Omalizumab, I don't think really anybody knows. There is some data on, mechanistically how it works, and you can make conclusions based on that in terms of timing of when to start and when to stop. But really, we just need a lot more data on that. And I also put the cost issue in the cons section as well. Very, very expensive, and so the question is it worth it? And then also figuring out who gets it. How do we figure out which patients would truly benefit and which ones wouldn't, particularly because of the cost issue. It's not something we can just throw around. So those are questions that are still out there. The concept of multifood or simultaneous multifood OIT is very interesting. The rationale for that is that 30% of food allergic kids have at least one food allergy. And there is data to show that people with multiple food allergies have a worse quality of life and may suffer from dietary deficiencies. I don't think any of us would argue that. So couple of studies, really only a couple of studies looking at this topic. So this one was published earlier this year from the Stanford group, looking at multi-food immunotherapy. This trial involved 40 patients, both children and adults. They wanted peanut allergic people, and then either monoallergic or peanut allergic plus an allergy to at least one other food. And the foods they were looking at were tree nuts, sesame seed, milk, and egg. One of the strengths of this study, they did confirm the food allergies with challenges before. With every one of the allergies, they challenged. That was Gary's phone. Anybody else want to?
60s. That's our best women's group.
So they randomized, or no, I'm sorry, they didn't randomize. They put 15 people into the peanut monoallergic group and 25 into the multi-food group. So they had a fairly equal distribution of people who had 2 foods in their cocktail, 3, 4, and 5. They tried to look fairly evenly at the different, combinations or multiples of foods. And they were shooting for a maintenance dose of 4,000 milligrams per food. So eventually could make it up to 20,000 milligrams of cumulative protein if they were on the full program. So they found that all of these subjects, all 40, reached their primary endpoint, which was a 10-fold increase in reaction threshold. But they did find that the multi-food groups were slower, 14 weeks slower on average compared to the mono immunotherapy group. And that's because if you have, the more food allergies you have, they were starting with smaller amounts of protein, so it took longer for them to build up to the dose. And as far as adverse reactions, they actually didn't find any significant difference between the multi-food group and the monotherapy group. No serious events, and if anything happened, it was mostly mild. For whatever it's worth, they found that the peanut IgE did not change in either group, but the peanut IgG4 did go up.
How did they administer these? They mix the flours together and you just got all of them at once?
Yes. Exactly. So this is the same group, published almost around the same time. They did basically the same thing as the previous study, except this time they added Omalizumab. They wanted to see if Omalizumab would make any difference. So this study, a little bit smaller, they had 25 subjects, and these were all children. And they were looking at basically the same foods minus the egg. Like the other studies with Omalizumab, they did an 8-week pretreatment, total of 16 weeks. They began the desensitization on week 9. The maintenance dose they were targeting, 4,000 milligrams per allergen. And they found that of the 25 subjects, I'm sorry, I forgot to mention, but they did the same thing where they tried to evenly distribute the groups in terms of the number of allergens, so 2, 4, 5, and 6. And they found that almost 3/4 of all the patients reached a full 1250 milligram dose on day 1. So basically, more than a quarter of their maintenance dose they got to on day 1, and it's different from the previous study, probably because of Omalizumab. The median time to reach maintenance dose amongst the whole group was 18 weeks. Even with the group that had 5 or 6 allergens, that's still pretty fast. And they estimated, and the authors estimated approximately 67 weeks faster with Omalizumab compared to without, in terms of reaching maintenance. 94% of the reactions were mild, GI, itching, ENT symptoms. And in this group, they found that the skin test went down by a fair amount, 8 millimeters. And they did see the increase in peanut IgG4, no change in peanut IgG. So this is not. This is actually a study that will be relevant to us because we're going to be one of the centers for this. This is the Multifood the MTAX study. It's a phase 2 multi-center OIT trial with Omalizumab and Multifood allergic patients. And they're going to do something similar to the previous study, but they're also going to look at the tolerance aspect as well. So it's going to be a 36-week trial, and Asthma Inc. and Virginia Mason will be collectively one of the sites involved in this study. It was supposed to start this fall, but it's probably going to be early 2015 before we start recruiting. This is going to enroll 60 subjects total, children and adults. And we're going to look at up to 5 food allergies. And this study is going to be a lot of the same ones as the previous ones: milk, wheat, egg, soy, nuts, and a few fish and shellfish as well. So they're going to get 8 weeks of Omalizumab, 16 total. Initiate the rush desensitization at week 8. This one's going to be a little lower dose maintenance, 2,000 milligrams. And their test for tolerance, the design is going to be at 30 weeks. They're going to go through a challenge, and if they are, if they're not reactive, in other words, if they pass, that group will be randomized to one of 3 arms. They're going to either continue what they're doing, they're going to decrease to a lower dose, and that'll be 300 milligrams per food, or they're going to get placebo, and then they're going to be challenged at week 36. So, interesting design. We'll see how that
What's
goes.
going to happen to the ones who failed the challenge at that point?
I believe they're going to keep going. They will keep.
And there'll be immunologic studies to look at T cells. And
all of them. I want to highlight ARC, Allergen Research Corporation. They've got an ongoing open-label phase 2 trial with peanut using cPNA, characterized peanut allergen. It's basically a modified peanut protein, and the benefit of using a product like this as opposed to peanut flour or peanut extract is that you have guaranteed dosing, potency, purity, and stability. So you basically have control over what you're giving. That's the rationale behind it. This trial that's ongoing has 50 subjects, children and young adults. And currently there are 8 centers in the United States that are involved in the clinical side. We're not one of them, but Benaroya is responsible for doing all the mechanistic studies for all the people enrolled in this study, so it is something that we're involved in. Right now there's active discussion with ARC for Seattle to become one of the centers for the phase 3 studies, and I think that's going to happen. So that probably will, I think, be in 2015 sometime. And as far as the company itself, they have plans to roll out this characterized product for multiple foods: milk, egg, soy, wheat, seafood, and tree nuts. And they have a plan to roll these products out by stages over the next 6 or 7 years. So I presume with that, hopefully we'll become fairly active with that through CFAC. So I'm going to switch to Slit now. Slit is allergen extract placed under the tongue, then you either spit it or swallow it. Doses are much smaller. I believe I had a typo in the handouts. It says 10 grams, but it's actually supposed to be less than 10 milligrams per day. It does appear to be successful in desensitizing people with fewer side effects, but the major gripe with Slit is that it seems to be less effective than OIT. And I just put a couple of references here for various foods that have been reported to be treatable with Slit. Mechanistically with Slit, very similar, except you're utilizing, similar to OIT, except you're utilizing the mucosal Langerhans cells, which then lead to the same sort of immunomodulatory effects through upregulation of Tregs. So I'm going to point out a couple of recent studies on Slit over the last 5 years. This one, kind of an interesting design looking at milk allergy and actually comparing Slit to OIT. So this is a 60-week trial, 30 kids, and they did confirm the milk allergy for all of their participants through baseline challenges. Every single subject escalated through Slit. And after a certain point, they were then randomized to either continue Slit at 7 grams per day or start OIT using either a high dose amount of milk protein, which is the 2,000 milligram A group, or the low dose, which is the 1,000 milligram B group. So they had these 3 arms that eventually split off. And they challenged everybody at 60 weeks to 8 grams, and they found that in the Slit-only group, only 1 out of 10 passed. In the low-dose Slit OIT group, 6 out of 10 passed the challenge. And in the high-dose Slit OIT group, 8 out of 10 passed. So it seems that OIT at higher doses appears to be the most effective means. And those who actually passed, these 15 kids, they were re-challenged. So they were told to stop their therapy at 60 weeks, and then they came back, I believe, at 1 and 6 weeks off therapy, but I'll just highlight the 6-week challenge. So 6 weeks off therapy, these people all came back and got challenged. And they found that 3 people from the low-dose OIT group and 3 from the high-dose OIT group regained their sensitivity. And the remaining subjects from this group were considered tolerant. How long that lasted, I don't know. They didn't report any follow-up data on it. But that did include the one person who got Slit only. So that's interesting. But I don't know if you can really make any significant conclusions from that. But it is what it is. The overall reaction rate between the groups was pretty similar, but the OIT groups, particularly the high-dose OIT groups, did have a higher rate of systemic reactions. So that's consistent with what we know about OIT.
David, was there any positive effect of being on Slit as far as reducing reactions during OIT? In other words, versus just doing OIT? You know?
Interesting question. I don't recall that there was any specific comment or data on that.
And the other thing that strikes me with all of these, when we do oral challenges in the office, the usual mantra is you need 10 to 15 grams of the food to say you're good, and yet these are much lower doses. Maybe that's why we're not achieving tolerance.
Well, they're not. I mean, that's an inherent difficulty with all of these studies, is they're not using real-world standards, but what they're using is, when they do the baseline challenges, they're using that as their benchmark. So they're trying to shoot. Most of these studies, one of the either primary or secondary endpoints is to reach for a tenfold increase in reaction threshold, which for some of these people, a lot of these people, is not that much.
Yeah.
So this is a Slit study for peanut allergy. This one is from Kim from 2011. 18 kids, 6 months dose escalation and 6 months maintenance. The active group tolerated 20 times more peanut protein than the placebo group at 12 months. So there was a positive effect in this study. Symptoms. More than 10% of all doses led to symptoms for most of the trial. I'm going to move on in the interest of time here. Slit, another Slit study. This one looked at 40 subjects, children and adults. They did 44 weeks of Slit and found that 70% of this group were positive responders, as opposed to 15% of the placebo group. That means either a tenfold increase in reaction threshold or the ability to consume 5 grams. The daily amount that they consumed increased from 3.5 milligrams at the beginning to 500 milligrams at 44 weeks to 1,000 milligrams at 60 weeks. So significant, improvement. A lot of, in terms of the total number of doses, up to 40% of the doses led to reactions, but like the others, most of them mild. In this study, they found an increased peanut IgE at the times that they assessed. Decreased IgG4, that was an interesting paradox, and basophil reactivity at 44 weeks, and no changes in the skin test. So that's Slit. I'm going to talk about patch for just a minute here. This is another issue that I think we can all resonate with because we've been very active in this in terms of investigating this modality here in Seattle. This is what the Viaskin patch looks like, made by DBB. That's sort of, what it actually looks like. The patch has the antigen in a dry form. They've created this patch to include a condensation chamber, so the idea is when you put it on, the antigen solubilizes and it more easily penetrates the skin. So the idea with the Viaskin patch is that you apply it on the skin for 24 hours, take it off, put another one on, and that you're doing that mechanistically with patch, again utilizing Langerhans cells in the skin to upregulate Tregs. This time through the help of CTLA-4, you have basically the same proposed downstream effects in terms of shutting down the allergic response. A couple of trials here. This is a pilot study for cow's milk patch in 2010. Very small study, 18 kids. These kids got this study is 48-hour patches 3 times a week. And they found that the cumulative reactive dose was more than 10 times the reactive dose at baseline, so they were able to tolerate 23.6 mLs at 3 months as opposed to 1.8 at baseline. So a significant increase in a short period of time, and the adverse reactions were mainly localized, redness, eczema. This was one of the earliest DBV peanut patch studies. It was a phase 1 safety trial here in the U.S. Very short. It was only a 2-week trial, 80 subjects with peanut allergy, 20 with placebo, basically trying to figure out what doses were tolerated. And they found in this study that 250 micrograms was well tolerated in children and 500 micrograms in adolescents and adults. And like the previous study, most of the side effects were localized skin reactions, nothing severe, and no need for epinephrine in this short study. The ERA Child is an ongoing study. It's a 3-year phase 2A study that's exclusively in France, 54 kids. They were treated only with the Viaskin 100 microgram patch. They were getting challenges every 6 months on the patch. And as of most recent data, they have a challenge out to 18 months on these subjects, and they found that approximately 70% were hitting their endpoint, which was a 10-fold increase in reaction threshold. So a positive effect after 18 months. And safety is consistent with some of the phase 1 data that I just talked about. Vipes, we're all very familiar with this. This is still ongoing in terms of the follow-up phase. It's a phase 2B study at this point, or it was a phase 2B study, 22 centers in the U.S. and around in Europe, 220 subjects, both kids and adults, and 9 of them were here at Asthma Inc. The arms were looking at low, medium, and high dose peanut patch, 50, 100, and 250 micrograms, versus placebo. And we know that the highest dose, the 250 microgram patch, met its endpoint of a 10-fold increase in cumulative reactive dose compared to baseline. And I just wanted to point out that alongside this, COFAR was doing their own study with DBV. That's still ongoing. It's a phase 2 study looking at 100 mics and 250 mics. They have 75 subjects, and it's going to be over 30 months.
There are no placebo patches. Some of these people might be naturally changing their threshold.
Yeah, right. That's always a question, no matter what modality we're looking at. Right. This is where we are now with this cohort. So this is the open label follow-up study. They were able to get 170 out of the 220 from the VIPES cohort, and they're all being treated with 250 micrograms Viaskin patch, and that's going to go out an additional 2 years, out to 36 months. They want to look at safety and also sustained unresponsive. So they're going to have, at 36 months, they're going to go off treatment for 2 months, and they're going to come back in and get challenged. And there's the phase 3 trials for this product are going to start. It was supposed to start right around now, but I don't think that it's recruiting yet, so it's probably going to be early 2015 before that happens.
Have they thought about putting these kids on the oral then after they do the patch? You know,
that's a good idea, sort of a hybrid. I don't know that anybody's thinking about it. People have probably thought of it, but I don't think there's.
I mean, it's sort of like the kids that pass the peanut challenge. It's almost like then they should just start eating like a Reese's peanut butter cup every day,
right.
You know, and then just maintain their tolerance.
Yeah.
And whether they ever eat a peanut butter sandwich, but just to maintain it.
Right. The MILES study, also something we're going to be involved with here. It's going to start out as a multi-center phase 1, phase 2 trial looking at Viaskin milk patch. They're going to look at low, medium, and high dose patches. It's going to be a 1-year treatment with 150 kids. Asthma is going to be one of the 10 sites in the U.S. recruiting. Right now, I think only the Canadian sites are recruiting, but I think this is going to start pretty soon. And Smiley, this has not started yet, but it's a trial that sort of is initiated by CHOP and DBV. It's going to be a 9-month patch trial in 20 kids who have EOE, and they're going to, after 9 months of therapy, they're going to stop, and they're going to reintroduce milk into their diet in a controlled way, and they're going to get repeat biopsies after 2 months to see what's changed. That's supposed to start next year. Okay, so just a couple of slides to kind of summarize things. So we're seeing that these modalities, they are good at desensitizing a number of foods. But as we can see, only a very, very small subset seem to achieve tolerance or sustained unresponsiveness, which is kind of the ideal goal of all this. We are seeing immunomodulatory effects, decrease IgE, skin prick, Th2 signature, increase in IgG4 and Tregs, for whatever that's worth. If those are even the right markers to be looking at, that remains to be seen. But these therapies they do seem to make these changes. Safety is a concern. We're seeing that up to 20% fail the escalation phase and withdraw. Up to 20% never get to the full dose. Up to 25% can experience severe reactions during their protocols. These numbers have to do with OIT without Omalizumab. But having said that, most of the side effects are mild to moderate. We are seeing EOE, just a handful of reports on, but it is there. Slit appears to be safer, but it also appears to be less effective. Patch is showing some promise, but we need more data on that. I wanted to highlight this Cochrane review from 2012 on peanut OIT. Staggering. Only 1 out of 746 papers that met criteria for an unbiased trial. And based on the one study, that was the 2011 Varshney study on peanut OIT. Based on that one study, they could not recommend it. So basically, this is really far from real-world application. One of the issues for me, we've talked about it a few times here, the fact that all but one of these trials excludes severe anaphylaxis. So, and that to me is the main group that we're really trying to figure this out for. So it's hard to know exactly how to apply this data and these techniques without that information. There's just an extreme amount of heterogeneity from the trials that we have, but it's also important to point out that really in the last 2 years, the study designs are getting better. Many more randomized, blinded, placebo-controlled trials. More people are doing baseline challenges to confirm allergies. They're using larger cohorts, and they're also treating for longer too, which I think is going to be necessary to figure out how this works. So that's all I have. Yes.
I remember reading in England they were doing some open peanut powder trials as outpatients. And did you come across that in your reading at all? What the outcome of those studies were?
Is it Gideon Lack? Is he the one?
Compared to Israel, that one?
No, it was in England, and they were giving the kids outpatient doses of peanut powder, starting with like nanograms and then going up.
They're gonna. If I'm thinking of the data you're thinking about, I think they might actually be presenting it at the Quad AI in a couple months. I don't know, Lisa, if do you know something about?
Well, I was at the AAP meeting on the section on
Okay.
Allergy. They were talking about how that's going to be discussed at Quad AI.
Okay. Yeah.
Have they looked at patient satisfaction? Is it worth all the bother? Have they surveyed that?
Yeah, so there is a little bit of data on that. In terms of satisfaction, yeah, I mean it's better than doing nothing.
They would do it all over again.
Well, I think it depends on who you ask. The ones who are using epi and anaphylaxing, probably not. But I think just psychosocially, it's something that they're actually doing proactively. So I know personally from my patients, there's a high degree of buy-in. Whether we believe it or not is a different issue, but it's all we have right now. So there's a lot of buy-in.
Mary's point, I never thought about this before, but with aspirin desensitization, we accept the fact that you can't make people tolerant. We just take them through the protocol and say, take aspirin every day. Right. And although you want to get these people tolerant, for the moment you could just take peanut every day. And it's at least worth it if you don't forget that you went through the desensitization.
It's what are our expectations? What are our goals? If we're happy with desensitization, then we found it. It works. But I think most people are not satisfied with that.
Well, you at least protect people from life-threatening anaphylaxis.
Exactly.
The same thing you do with aspirin.
Well, they don't, because they're not taking the worst patients.
Well, that's another issue. They're afraid to take the worst patients.
I don't understand why more analyses are made because we take severe anaphylactic drug reactions or even alcohol all the time. We know that we have decades of experience that nobody's becomes tolerant, but it works very well. This clearly works. Treating with severe patients.
That's a good point.
I think it's also, there's been such a small number so far, maybe 30,000 at most patients. And, what's the risk? I mean, the risk of dying from your allergy is pretty small in this day and age, right? With peanut, I mean, the risk is, there was a report that you have a higher chance of dying from being struck by lightning.
You said 200 cases in a year.
That's the highest.
So when you, what's going to be the risk as this rolls out to more and more people? And what happens is you eat this peanut and then maybe you go out and play a basketball game. I mean, there's all kinds of cofactors, illness, activity, stress. So what's going to be, as it goes up to 100,000 to 200,000, is there going to be anaphylactic? Is there going to be a fatality, basically? So that is the biggest question, is are we actually increasing people's risk by putting them on
So your point you're making is you're stable, you take this amount of peanut every day, then you get a URI, your immunologic profile changes, and that dose is now dangerous for you.
Yeah.
Right. So actually,
that's what's happening.
One thing I should mention is that some of these authors that I pointed out, they built that into their protocol. So if they were told to report URIs, to the authors and to the investigators, and if they did, they built in a 50% dose reduction during the illness.
These are very highly motivated You know, they're talking to their doctor every day. I mean, as this rolls out into real world, there's going to be a lot more difficult, and it's going to be potentially more dangerous.
That's why the data from the England study looked so promising. They're just giving these kids every day, and they're not getting.
On Monday of the Academy meeting, Gideon Lab is giving the results.
Oh, there you go. So then the following Monday, you can tell me what they are.
Just kidding.
So you're going to tell us that we had that video issue today?
I'll go over. I'll stop chamber office before I remember to.
Thank you so much.
Are you in the garage downstairs?
What's up? Still working on that. I have to take the EPIC training the first 2 days in January when I get back.
And then you were thinking grandkids here at one point?
I just got an email on that today. I have to ask Kate when she really wants to take it. We just about to sign up with those 5 people got murdered.
No, I'm
Every
not.
time that happens, she's
I know that David Robinson.
to be honest, what's happening over there is, I don't know if this is a calculated effort or not to keep up.
I can pass this on to David. Oh, there's. Thank you. All right. Thank you very much. Have a good day.
So I'm trying to, find definitely find some things that work. I don't make that contract.
So what's going to, hey, very nice.
Well done.
Feel free.
I will come up with a list of days that
But it could be, that sound like a
we got much less, but I guess before starting to offer you some
I don't
days.
know if
So
they
yeah,
have
it's
to
still
sign.
there,
I think you go online and put that password in.
Oh, that's not soon were you thinking of starting?
And you might have to call the guy that got that for that's a Dr. Eric. So that it won't budge out to you. We will hold my
Hawaii vacation, I think the 18th of this month.
Okay.
I'll try and get her these definite dates for January, February, and March.
Yeah. You're trying to pick up right? I'm just
Any food left for the road, guys?
Yes, sir. Do you want the rest of the fruit?
Today.
This would be good for your cold.
Yeah. Take one of these.
There's a cream cheese in here if there's a bag of left.
So what is your what is your territory now?
It's
You just
downtown
still?
Seattle,
Oh, downtown.
all the way toward Longview, Aberdeen, and then out to the peninsula.
But you don't have Northgate then.
I'm cut off like right down here.
That's,
Yeah.
I'm Christy, by the way.
Hi, Chris. Oh yeah, my friend Adeline. You know Adeline?
Yes.
Yeah, she's like, if you see Christy, say hi to her.
Yeah, she's